What if an antibiotic kills almost every bacterium in an infection, but the few that survive become the start of a much bigger problem? That is the basic idea behind antibiotic resistance. And the first thing to understand is this: your body does not become resistant to antibiotics. The bacteria do. Bacteria do not think, plan, or study a medicine. But they reproduce extremely quickly, and every new generation can contain tiny genetic differences. Most of those differences do nothing useful. Occasionally, however, one bacterium happens to carry a mutation that helps it survive a particular antibiotic. Some bacteria can also pick up resistance genes from other bacteria. Now imagine an infection containing millions of bacteria. Before treatment, most are vulnerable to the antibiotic, while a small number may already have some protection. When the correct antibiotic reaches them, it attacks a specific weakness. Depending on the drug, it may damage the bacterial cell wall, block the machinery that makes proteins, or interfere with another process the bacteria need to live. The vulnerable bacteria die or stop growing. That is good, because this is how antibiotics save lives. But the drug also creates a powerful filter. The bacteria that cannot handle it disappear. Any resistant survivors face much less competition for space and nutrients. They can multiply and become a larger part of the population. The antibiotic did not teach them how to survive. It removed the bacteria that could not. This is natural selection happening at high speed. And bacteria have another advantage. They do not always have to wait for a lucky mutation to appear in their own descendants. Some can share useful genetic instructions with other bacteria. One important method is called conjugation. Two bacterial cells make direct contact, and one can pass a small piece of DNA called a plasmid to the other. A plasmid is like an extra instruction card stored outside the bacterium's main chromosome. It may carry a gene that helps destroy an antibiotic, pump the drug out of the cell, or change the target that the drug normally attacks. The USB file transfer comparison is not perfect, but it gives you the basic picture. One bacterium can pass useful survival information to another. The receiving bacterium may then copy that DNA when it divides, and in some cases pass it onward. This does not mean that one survivor always turns an entire infection resistant in a few hours. The speed depends on the bacterial species, the resistance gene, the drug, and the environment. But gene transfer can allow resistance to spread much faster than if every bacterial family had to wait for the same mutation by chance. So why can resistance move faster than new antibiotics are created? Bacteria can produce new generations in minutes or hours. Huge bacterial populations create countless opportunities for mutations and gene exchange. A new antibiotic, meanwhile, may take years of research, testing, approval, and manufacturing before doctors can use it safely. That does not mean antibiotics are the enemy. They are essential medicines, and refusing one when it is truly needed can be dangerous. The problem is unnecessary or incorrect use, because every exposure gives bacteria another opportunity to be selected. Taking an antibiotic for a viral cold will not kill the virus. Using someone else's medicine, saving tablets for later, changing the dose, or stopping or extending treatment without medical advice can also make treatment less effective and increase risk. The safest rule is simple. Use antibiotics only when they are prescribed for a bacterial infection, and take the exact drug, dose, and duration recommended by the healthcare professional treating you. Do not share them, and do not change the plan on your own. Hospitals and laboratories also fight resistance by identifying the bacterium, checking which drugs still work, preventing infections from spreading, and choosing the narrowest effective antibiotic whenever possible. Vaccination, hand hygiene, safe food handling, and infection control matter too, because every infection prevented is one less chance for antibiotics to be used. Antibiotic resistance is not a story about bacteria becoming clever. It is a story about variation, survival, reproduction, and shared DNA. The medicine removes the vulnerable bacteria, the resistant ones survive. Then they multiply, spread, and sometimes share the genes that protected them. That is how a treatment designed to defeat an infection can also create pressure that favors the bacteria best able to resist it. Antibiotics are still among the most important tools in medicine. The challenge is to use them carefully enough that they keep working when we truly need them.